Hi, my name is Alex Lanai. I'm a computational technician in the Frankel Lab here at MIT, part of the Neuralinks and Answer ALS consortia, and I'm going to talk to you about reproducible science. So more specifically, with our computational pipelines, we'd like to be able to maximize the reproducibility of those pipelines in our computational experiments as part of the consortium. And also, wrapped in with that is this idea that we'd like to make it easy to run large-scale analyses for the researchers that are part of the consortium, particularly the sort of analyses that we'd think of as standard. So I'm going to structure this presentation as first sort of like raising a set of problems that you may be familiar with, and talking about later the solutions that we came up with for those problems. And this is the first of a set of tutorials about how to use the infrastructure that we've built here at the Frankel Lab. And the later ones will be more hands-on, and this one is more to explain our entire approach to all of these problems and our hopeful solutions. So the problem that we face is that computational pipelines are brittle. What does this mean? So the brittleness of a computational pipeline means that it might work on one environment, but then when you try to go to transport it to another environment, oftentimes it doesn't. And there are a variety of reasons this might occur. Oftentimes environmental variables and just the operating system that a computational pipeline is built on and runs in, the pipeline might be very dependent on those environmental variables, meaning that once the environment is changed, we find that the pipeline doesn't work. And this is obviously a problem for reproducibility. It's also a problem for transparency, and it might question the validity of your results if other researchers can't necessarily reproduce your analysis the same way you might expect in a wet lab. If computational analysis can't be reproduced and people come up with different results, then this is a problem. So introducing Docker. So Docker is a technology that's was invented a couple of years ago. It's been pretty widely adopted in industry, and now is starting to have some major impact on science. And Docker is a technology which containerizes programs. So what does that mean? To containerize a program means that you take a program and you wrap it with the operating system and environment that it runs on inside of what is metaphorically a container, but is really just an executable program that works on any environment. And this is extremely appealing because now you can take your pipeline, which works on one environment, and move it onto another environment. And since it is containerized, it will actually work. And the analogy to think of is a shipping container. Shipping containers made a dramatic impact on the shipping industry some number of years ago, because you could now take any good that could fit inside a shipping container, and you could wrap it in a shipping container. And now the shipping industry only needs to accept one interface for moving goods across the world. It no longer needs to deal with having to consider the different ways of moving different goods. So we've built Dockerized Galaxy. So you may be familiar with Galaxy. Galaxy is a program for visually wrapping the command line and running analyses visually. And we've built a couple of features into our Galaxy. So I'll walk through them. So first, you can securely upload data. We contributed this back to the Galaxy source code, the ability to sort of meet NIH guidelines around data security, which is very important to us. We have tools which provide sort of reproducible data analysis. Each of them is containerized. So although the Galaxy itself is a Docker container, each of the tools are individual Docker containers, meaning that each of the computational pipelines that you could run are themselves these containers that can run anywhere and have this sort of standard interface with computational platforms. We provide workflows, which is the Galaxy term for pipelines. And we also have these histories, which are these essentially sequential data transformation trackers, which tell you what data you had and how you got from one data set to the next data set. And this provides a good record, a history of what analysis was performed, which we can later point people to once you can publish Galaxy histories. And then finally, we have this shared data icon, which provides you the ability to share data across the consortium, but at a very granular level. So there may be some data that you're ready to share with some researchers in the consortium, but not others. And you can decide which groups that you want to share your specific data with, and you can do this on a file-by-file level. This provides this entire platform provides a way for researchers to not necessarily need to worry about the computational environment they're running their tools in. They just see Top Hat or bow tie, and they run it with the parameters that they want to run it with, and we take care of the rest. And it's at galaxy.neuralinks.org. You can go check it out now. And the rest of the tutorials in this series will explain how exactly to use it. So this is a quick image of our AttackSeq pipeline, which is the first pipeline that we've set up here at the Frankel Lab. And this is an epigenomics pipeline. As you can see, we take our raw reads, we filter out the mitochondrial reads, we then sort the BAM file. We, on the one hand, convert it into a bigwig file, but mostly we call peaks with max2. And then later we might map those peaks that max2 gave us to known genes that are in the neighborhood. And then we then plot those, the distribution of sort of mappings. And this is obviously a very appealing interface to looking at a computational program because it's pretty unambiguous, it's pretty simple, and it's very visual, which is very appealing as opposed to having to sort of finick with the command line, and knowing that what you're doing relies on very specific versions of software packages and potential sort of environment variables which might not be common across operating systems and environments. So that concludes this first talk. I want to sort of leave you with the idea that reproducible science is important, that it's a real problem, and it's important to this consortium, and we achieve it through the sort of union of Galaxy and Docker, which we're running on Microsoft Azure. Thanks very much, and I look forward to explaining this in more detail in the next talk.